Hey guys, hello everyone and welcome to the channel. So welcome to yet another video of single short video series. In this series we are trying to do topics which are important from the exam point of view, and we are trying to finish it up in a single video. This video and this topic was most asked topic. This is Bio-inorganic Chemistry, and I hope that this video will help you in your exam for sure. So let's talk about Bio-inorganic Chemistry. In this particular video I'm going to take most important proteins, most important complexes, most important molecules which actually you have to study in Bio-inorganic Chemistry. I'm omitting the parts which contain the benefits or the uses of like different metals in the biological world. So those tiny things I'm not going to include in this video, just important portion from the most important molecules of Bio-inorganic Chemistry are going to be covered, right? So now before starting it, before going into the detail of this video, if you are new to this channel and if you have not subscribed the channel, you're watching the video, you are liking the content, please do consider subscribing it. It gives me motivation to make much more videos for you guys. Now without wasting any time, let's continue with our video. Okay, so the first molecule which we are going to discuss is cubane. So cubane has a molecular formula of C8H8. It's a synthetic hydrocarbon molecule. It consists of 8 carbon, okay, and the atoms are arranged in the corner of cube and with the hydrogen attached to each other. So why I am talking about cubane? Because we have to talk about ferredoxins, okay. So ferredoxins are small proteins that contain iron and sulfur atoms, and these are organized as an iron sulfur cluster. Now these groups are non-heme groups. Understand that these does not contain heme group, and which affects the electron transfer in the plant. So they are electron transfer proteins in the plant and in some bacteria, and does the same biological function as what cytochrome-c does or the cytochromes does in animals. So they are kind of cytochromes for the plant and bacteria. Now these biological capacitors, okay, can accept or discharge electrons, the effect being change in the electronic oxidation state from +2 to +3 of the iron atom. Thus ferredoxin act as an electron transfer agent in the biological redox reaction. Now they are called capacitors because they can contain the charges. You know what is capacitor is? In physics we study capacitor, which is put in the circuit, and it basically captures or it basically stores the charges. In the same way ferredoxin also does the same thing. They store charges by changing their oxidation state from +2 to +3. So this is a typical electron transfer mechanism for ferredoxin. You can see that how the oxidation state of the ferredoxin is changing. So the formal oxidation number of the iron can be 2 iron in +3 state and 2 iron in +2 state, whereas it can be also in 1 in +3 and 3 in +2 state in the low potential ferredoxin, where the charges are less. The oxidation number of the iron ions in the high potential ferredoxin can increase, and there the number of iron having +3 charge will be more, right? So that's how they transfer electron. Now there is one more type of ferredoxin, which is Fe2S2. Now this type of ferredoxin has the structure which is shown to you on the screen. It also has similar properties, just the structure is different over here. The next thing which we are going to discuss is cytochromes. So cytochromes are membrane bound heme proteins. Understand that these are heme proteins, that means they contain heme group. This is the heme group you can see on the screen. This is called a heme group, right? Now it contains 4 heme group as a cofactor and carry out electron transport. Most of them contain iron either in +2 state or in +3 state at their active sites. They are found in the mitochondrial inner membrane and the endoplasmic reticulum of eukaryotes, in the chloroplast of the plants, and in photosynthetic microorganism, and also in some bacteria. Now cytochromes are classified in three forms: cytochrome A, cytochrome B, and cytochrome C. They contains 3 different types of heme: heme A, heme B, and heme C. Now in cytochrome the iron is hexa-coordinated, that means it has 6 coordination number. The 4 coordination sites are occupied by 4 nitrogen of the pyrrole rings of the heme group, and the remaining are occupied by usually histidine and cysteine residues, okay. So basically if you want to see it, it's shown over here. The iron is tetra-coordinatedly shown over here, which is like iron is surrounded by 4 nitrogen of the heme group. On the upside it will be attached with histidine, and on the downside it will be attached with cysteine, right? Cysteine. So yeah, CYS, right? So next thing is, depending upon the ligand, the redox potential of a given cytochrome can be tailored to meet a specific need of electron transfer. Potential are such that electron flow is always from cytochrome B to cytochrome C, then to cytochrome A, and then oxygen is transferred. In the mitochondrial electron transfer chain, cytochrome C accepts an electron from the cytochrome C1 and then transfers it to the cytochrome C oxidase. Ultimately, the electron is used in the 4-electron reduction of O2. Only cytochrome A has the ability to bind to oxygen and to reduce it. Remember, these are very important. Each point in this slide is very important. That's why I'm making these videos. You can pause the video and take the screenshots. You are totally allowed to do that, right? Next is that the Cn minus ion can bind strongly to the 6th coordinated site and stabilize iron plus 3 in cytochrome A. This makes cytochrome A to stop functioning in the electron transfer reaction. So this point is all about the functioning of cytochrome A. Cytochrome C is a heme protein and coordination number of iron is 4 from the heme and 6 due to histidine and cysteine, which we have already shown, like I have already shown you how it is. Now it is a redox protein and act as an electron carrier. Only cytochrome A has ability to bind to oxygen. Cytochrome C does not act as an oxygen carrier, nor it can store oxygen, right? So these are just some of the summary from the cytochromes. Next, let's talk about carbonic anhydrase. So an enzyme present in the red blood cell is called carbonic anhydrase. It aids in the conversion of carbon dioxide to carbonic acid and bicarbonate ion. When the red blood cell reaches the lungs, the same enzyme helps to convert the bicarbonate ion back to carbon dioxide, which we breathe out. So this carbonic anhydrase is a very important enzyme which carries out the excess or used carbon dioxide from your body, and it takes it till the lungs so that you breathe that out. The active site of the carboxy anhydrase contains tetrahedrally coordinated zinc plus 2 ion, and it has coordination to 3 histidine residues and 1 water molecule. This is how the active site of carbonic anhydrase looks like. Its function is to catalyze the conversion of carbon dioxide to bicarbonate. So carbon dioxide is converted into bicarbonate by the help of carbonic anhydrase. How the mechanism follows for the conversion of carbon dioxide to bicarbonate. So the step 1 is deprotonation of coordinated water molecule. Okay, so the step 1 is there is a deprotonation. You can see in the first step this hydrogen ion is going to get removed, and this is a deprotonation step. This is the crucial step. The zinc ion bound to the water is more acidic than the free H2O and loses a proton easily, because here the water is more acidic. It is bound to the metal in the +2 state. That's why it can simply lose one of the hydrogen ion. Now thus it forms zinc bound hydroxyl group. Now here I have zinc bound hydroxyl group, and it carries out nucleophilic attack on the carbon dioxide. So carbon dioxide can it can form a nucleophilic attack on it, and it can form a carboxylic acid group attached to the metal. Now it is a nucleophilic addition on carbon dioxide. This should be very much clear to you. In the next step, there is a displacement of OCO2H by the water molecule. So here you can see that water is going to come and it is going to remove this whole thing. So what is going to get removed? That is HCO3 minus is going to get removed, and water is going to come back. So the cycle is complete like that. Let's talk about hemocyanin now. So hemocyanin is a protein which transports oxygen in the bodies of some invertebrates. Their active center has 2 copper atoms which reversibly bind a single oxygen molecule. Unlike the hemoglobin in the red blood cell found in vertebrates, hemocyanin are not bound to the blood cells. Alright. Hemocyanin is respiratory protein containing 2 copper center at the active site. It is a dioxygen carrier suspended in the hemolymph, that is blood of most of the molluscs and arthropods. It contains no heme, so it does not have any heme group. The deoxy form contains copper in +1 ion and is colorless, whereas the oxy form contains copper in +2 ion and is blue in color. Now in the deoxy form, each copper plus is coordinated to 3 histidine residues. Side-on bridging coordination occurs with the dioxygen in the oxy form. So this is deoxy form and this is oxy form. You can clearly see that the copper +1 is attached with 3 histidine units here. Here also this is copper attached to 3 histidine units. In the oxy form, they are going to form a side-on bridging with the dioxygen, and that's how the oxy form of your like hemocyanin is going to be formed, right? So basically the active site contains this unit, okay, and this is attached to 3 histidine units. Okay, so copper 2 has D9 configuration. We all know that D9 configuration is very much active for joint distortion. The unpaired electron in both copper plus 2 ion couple by bond interaction through the bridging peroxy ligand, and hence oxyhemocyanin is diamagnetic. Okay, these are points which you have to remember. Spectroscopic evidences of the above oxyhemocyanin structure are given. Like Raman spectroscopy indicates that symmetric binding and it rules out the mononuclear peroxy compound. And oxyhemocyanin is EPR silent, that means it is EPR inactive, which means that it is diamagnetic and there is no unpaired electron present in the molecule. Let's talk about rubredoxins. So rubredoxins are low molecular weight iron containing bacterial protein. They are involved in the electron transfer. Sometimes rubredoxins are also classified as iron sulfur proteins. However, in contrast to iron sulfur proteins, rubredoxins do not contain inorganic sulfide. Okay, this is very important that rubredoxin does not have inorganic sulfide in them. Rubredoxins active site contain an iron ion either in +2 or in +3 state, which is coordinated by the sulfurs of the 4 cysteine residues, forming an almost regular tetrahedron structure. Now, it is involved in the electron transfer in the respiration, in the mitochondrial and photosynthesis plant. In the mitochondria, it accepts electron from ubiquinol and transfers electron to the heme iron of the cytochrome-c, whereas in the chloroplast, it accepts electron from plastoquinol and transfers electron to the heme iron of the cytochrome-f. Now let's talk about hemerythrin. So hemerythrin is an iron containing non-heme oligomeric protein which transports oxygen in the marine invertebrates. It's a respiratory protein, that means it transfers oxygen. Remember, respiratory protein means some proteins which transfers oxygen. The monomer of hemerythrin is myohemerythrin, which is present in the muscles of marine invertebrates and stores dioxygen. Just like hemoglobin and myoglobin, they are working in the marine invertebrates. So just like hemoglobin, there is hemerythrin, and just like myoglobin, it is myohemerythrin. Deoxyform are colorless, whereas oxyform are violet pink in color. Active site, so the oxygen binding site is a binuclear iron center. Deoxyhemerythrin contains 2 high spin ferrous ions bridged by hydroxyl group. The iron ion in the coordinated to the protein through the carboxylate side chain of 1 glutamate, 1 aspartate and 5 histidine amino acid residues. This is how the active sites look like. So you can see that 2 iron atoms are attached to each other or are bridged to each other through the hydroxyl group, and this side you have aspartate and this side you have glutamate, and on one side you have 3 histidine units and on the other side you have 2 histidine units. So one iron is hexa coordinated, whereas other one is penta coordinated. So this iron is hexa 1 2 3 4 4 5 and 6 coordination this side, whereas this one is 1 2 3 and 4 and 5, so penta coordinated. So the penta coordinated iron binds to the triplet dioxygen and oxidized to iron +3. Then the hydrogen of the hydroxy group is transferred into the peroxide group. Now the second iron that is in the +2 is oxidized to +3. In this process, an oxo bridge is formed between the iron ions. Unlike in hemoglobin, there is no cooperative effect observed in the case of hemerythrin. Its affinity towards CO is less than O2. Let's now talk about the most important proteins. These are hemoglobin and myoglobin. So first of all, let's talk about that. What are heme proteins? So heme proteins are basically conjugated proteins with heme as the prosthetic group in their structure. Heme containing proteins are like hemoglobin, myoglobin and cytochromes. Heme containing enzymes are like catalase, peroxidase or tryptophan dioxygenase or tryptophan pyrrolase. Now, what is hemoglobin? So hemoglobin is the major heme protein of human body. Hemoglobin chemically is a conjugated protein, and in hemoglobin there are 2 parts. One is heme part, which is prosthetic group, and the other one is globin part, which is protein part. Hemoglobin can be named, like it has been named based upon heme plus globin, right? Hemoglobin is a red color pigment. It is located inside the red blood cell. This is the absorption band of oxyhemoglobin, and deoxyhemoglobin is shown over here. There are certain terminologies which are associated with hemoglobin, like hemoprotein means heme is there in that particular protein. Chromoprotein means it has a particular color, that is red. Metalloprotein because it contains a metal, that is iron. Respiratory protein because it connects the respiration process and it acts as a oxygen transfer, right? And oxygen binding protein because it binds with molecular oxygen and transports it. So these all terms are associated with hemoglobin. These all terms you will get to know when you will study hemoglobin. The structure of hemoglobin is that, like, basically it looks something like this. Iron containing pigment is your heme and it is attached to the protein which is globin, which is over here. Now heme is the iron porphyrin complex called iron protoporphyrin 9, and globin is a protein. This is the structure which you can see over here. Now iron in the heme has a +2 state and it is in the ferrous form in the reduced state. Iron +2 located centrally of the protoporphyrin ring system. So it's present in the center of it. Iron of the heme is hexavalent. 4 bonds are linked with nitrogen of the 4 parallel rings of the heme group. 5th bond is linked with the proximal histidine unit of the globin chain, that is globin, and 6th is bonded with the oxygen. So 6th side it will bind with the oxygen, right? Alright, so this is how the oxygen is binded to the heme group. So iron in +2 of the heme is linked to proximal histidine F8 and oxygen is linked to distal histidine E7, and thus to attain stability oxygen is bound to both heme and to globin both. So here you can see this is your dioxygen, it is bound to the heme also and to the globin part. So this is the distal histidine and this is coming from the globin part, and this is the proximal histidine. So iron is linked to the proximal histidine and then it is attached to the oxygen, and that dioxygen is attached to the distal histidine, right? So that's how the bonding happens. So in 3D this is how it looks. I'll just erase this so that you can see it clearly. And this is a 3D diagram of how iron binds, or how basically heme group binds with the dioxygen. Alright, there are certain features of oxygenation of hemoglobin. So oxygen binds to hemoglobin to form HbO2. Oxygen links to ferrous form of the iron of heme and non-enzymatically, loosely and reversibly. During oxygenation, one hemoglobin molecule with 4 heme can bind to 4 oxygen molecule. And during oxygenation, ferrous of heme is not oxidized to ferric. Oxygenation of hemoglobin causes considerable structural conformational change in the globin subunit. You can see this is the deoxygenated part where both these heme is domed, or you can say they are non-planar in the system, and this is your proximal histidine or the globin part. Now when it is oxygenated, there is a gap, or you can say there is this vent kind of arrangement of the heme group is basically becoming planar. So in the deoxygenated form, you can say that the heme group is non-planar, whereas in the oxygenated form, the heme group becomes planar, and that is because the repulsion created by this oxygen or dioxygen over here. And yeah, this is how it looks like. So that's the change in the structure of the heme from deoxygenated to the oxygenated hemoglobin. Here it is shown very clearly that how in deoxygenated or deoxyhemoglobin, how does it looks. It is little bit bent over here, whereas in the oxyhemoglobin, you can see that now it has become quite planar in structure. Now there are two forms of hemoglobin. One is called T form or tense form, and the other one is called R form or relaxed form. So what is T form? So deoxyhemoglobin is in the T form, that is in the tensed form. That's why it is bent, right? The iron, the heme group are bent here, and it binds with carbon dioxide H plus and other groups, whereas the oxyhemoglobin is in the R form, which is called the relaxed form, and where the iron and the heme groups are planar and it only binds with oxygen. Okay, T form has 8 salt bridges linked in between the dimer subunit, whereas in the R form the salt bridges are broken in between the dimer subunits during the oxygenation of hemoglobin. T form is more constrained form, whereas R form is less constrained form. T form has low affinity for oxygen, whereas R form has very high affinity for oxygen. T form of hemoglobin predominates in the low PO2, whereas R form of the hemoglobin predominates at a high PO2. That means when there is a less number of oxygen present in the blood, the hemoglobin will stay in the T form, whereas at the high when the high oxygen is there in the blood, then hemoglobin will be present in the R form. Alright, now let's talk about myoglobin. So myoglobin is unsuitable for oxygen transport but well suited for oxygen storage. What's the reason behind it? Let's look upon it. So this is percentage saturation and the gaseous pressure of oxygen. Now you can see that hemoglobin exists, the saturation, the percentage saturation is lower for hemoglobin, and you can see that for myoglobin the percentage saturation is quite high. This bracket or this region shows the reduced blood returning from the tissues, and this region shows the oxygenated blood leaving the lungs. So hemoglobin can act in both the ways, when it is leaving the tissues in that way and when it is oxygenated and it is leaving the lungs. So hemoglobin has two different parts, or you can say that it has two different regions or two different behavior. One when it is not having oxygen, and the other when it is having oxygen. Whereas in myoglobin, once it acts or once it captures oxygen, it does not lose it that easily. Okay, so that it is very good for keeping it or for storing it, whereas hemoglobin is not good at it initially, whereas later on at higher gaseous pressure it is good at storing it, but at lower gaseous pressure it cannot store oxygen. It has to leave the oxygen and give it to the cells and mitochondria. So myoglobin is a muscle cell and is oxygen storing heme protein. It is also called as reservoir of oxygen. Myoglobin does not allow oxygen to remain in the free state. Oxygen diffused in the muscle cell is used up in the oxidative phosphorylation. If oxygen remained unused in the cell, it immediately binds with myoglobin to form MbO2. Myoglobin is present within muscle cell, comes out in the blood after damage to the muscle cell. Myoglobin is found abnormally in the blood and urine of MI cases. Thus, elevated myoglobin level in the blood urine is a marker of myocardial damage. Let's talk about some differences between hemoglobin and myoglobin. So hemoglobin is an oxygen transport protein, whereas myoglobin is an oxygen storing protein. Hemoglobin is a tetrameric, has 4 heme group, whereas myoglobin is monomeric and has just 1 heme group, and it binds with 1 oxygen atom, whereas hemoglobin binds with 4 oxygen dioxygen atoms or dioxygen molecules. Hemoglobin are oxygenated at lungs, whereas myoglobin are oxygenated at the muscle cell cytosol. HbO2, that is oxygen bounded hemoglobin, unloads oxygen at tissues when PO2 is at 40 millimeter Hg or mmHg. Myoglobin or MbO2 unloads oxygen at the cell cytosol when the PO2 is at 5 mmHg to rapidly respiring cells. Now these values you don't have to remember, just you have to understand that what it does. ODC is sigmoidal in shape, so you don't have to worry about this point. Okay, that point is not that important. Hemoglobin has 574 amino acids. Molecular mass is quite very high, 67,000 daltons or 67 kilodaltons, whereas myoglobin is a smaller protein compared to hemoglobin. It has just 153 amino acids and the molecular mass is 17,200 daltons or 17.2 kilodaltons. Alright, that's all for this video, guys. Thank you so much for watching, and I hope whatever we discussed in the video was useful to you. I hope this video will definitely help you in the upcoming exam, and you will score questions. You will be able to do questions if they will be asked from the respective topics which we discussed in the video. Thank you so much for watching the video, and do give it a like if you like it, and do share with your friends, and also do subscribe to the channel if you have not subscribed it yet. Thank you so much for watching, and I will see you guys in the next one. Till then, have a great day. Bye bye. Take care. Hey guys, so I teach live on Unacademy Plus platform. Here I teach for the CSIR UGC NET category, and you can follow me over here for regular classes. You can access my free classes as well as my paid classes on this particular platform. The classes which are free, you can get that under the section of special classes, whereas in order to access my paid classes, paid live classes, you have to take Unacademy Plus subscription. So do make sure that you take the Unacademy Plus subscription to access all my paid classes, which are quite organized. The whole syllabus is being completed over there, and the classes are quite regular over there. So make sure that you take Unacademy Plus subscription by using my referral code, that is N_Huda. That's it for this. Thank you so much.